Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine being able to design a tiny, precise genetic switch, one that only flips on in exactly the cells you want it to, and nowhere else. No more off-target effects, no more guesswork, really. This week, we're diving into a really groundbreaking study that brings that future much, much closer. This isn't just about finding gene regulators anymore. It's about actually building them from scratch with incredibly specific, programmable control.
Yeah, and what's truly fascinating here, I think, is how this research tackles one of biology's really big challenges, the prediction and control of enhancer activity. For decades, we've been cataloging these vital DNA elements, right? But knowing exactly how to make them work, or crucially not work, in a specific cell type, that's remained stubbornly murky territory. So this deep dive really unpacks the causal DNA codes behind that control. It's pretty fundamental stuff.
All right, let's get into it then. Our sources today are excerpts from a truly landmark paper titled Designer Enhancers: Engineering Cell-Specific Gene Control in Blood. And our mission? Well, it's to show you how scientists are essentially writing the instruction manual for cell identity, one precisely engineered DNA sequence at a time, hopefully revealing insights that'll stick with you long after we're done here. So maybe we should start with the basics. What exactly are we talking about when we say enhancers?
Okay, good question. Think of them as tiny switches on the genome. They're specific bits of DNA that significantly boost gene transcription. That's the process of reading a gen to make a protein.
Right, so they turn the volume up on certain genes.
Exactly, and they're absolutely critical because they largely control a cell's identity. They're what tell a brain cell to act like a brain cell, or, a blood cell to be a blood cell. Without them, a cell wouldn't really know its job.
Okay, that makes sense. So if they're so important, why is predicting their activity so hard? What's the challenge been?
Well, that raises a really important point. Historically, we've used these maps, like from the ENCODE project you might have heard of, to find candidate enhancers. These are based on epigenomic signature.
Yes, chemical tags on the DNA.
Tags on the DNA and its associated proteins that kind of suggest potential activity. But, and this is the key thing, they don't guarantee it. Here's the kicker. Correlation does not equal function. It just doesn't.
Oh, okay.
So many of these so-called candidate enhancers, they simply fail when you actually test them in the lab. We've been, largely in the dark about the precise context-specific rules for their activation.
It sounds like having a map showing where light switches might be, but no idea how they're wired or which room they control.
That's a great analogy, actually, yeah.
And this is where the practical applications get really interesting, right?
Yeah.
Especially in something like blood development.
Oh, absolutely. Think about it. You have stem cells, and they need to branch out into dozens of different blood cell types. You've got platelets, red blood cells, all the different immune cells. It's complex.
So being able to engineer enhancers that only activate in one specific type, say, just in red blood cells.
Exactly. That could totally revolutionize things like gene therapy or CAR-RT manufacturing for cancer treatment. We're talking about achieving, unparalleled precision in medicine, making sure treatments only affect the cells we actually want them to.
Right, minimizing collateral damage.
Precisely. And to get that kind of precision, the fundamental gap this research tackles is that causal understanding. It's not just what DNA elements are there, but which combinations, what spacing, even the orientation of these little transcription factor motifs.
Motifs being those short DNA sequences proteins bind to.
Yes, exactly, those TF binding sites. Which precise arrangement makes an enhancer active in, say, erythrocytes, your red blood cells, but totally silent in monocytes, which are a type of white blood cell? Without knowing these precise design rules, current gene therapies that use natural enhancers can be, frankly, a bit risky. Well, they might misfire, activate in the wrong cells, or they might even get silenced over time, losing their effect. So this study took a different approach. A bottom-up synthetic strategy. They basically built thousands and thousands of controlled variants to try and statistically figure out this complex motif grammar. And this lines up perfectly with this urgent need we have for safer, more potent ways to control gene expression specifically in certain cells.
Okay, so they decided to build them instead of just finding them. How on earth did they crack this incredibly complex code then? What was the scale?
It was massive. They didn't just look at a handful of examples, they built this huge library. 64,400 synthetic DNA constructs.
64,000.
Yep. Each one 270 base pairs long. So think of each construct as a tiny, custom-designed piece of DNA. Each one encoding really defined arrangements of binding sites for 18 different key hematopoietic transcription factors, the proteins important for blood development.
Wow. It's like creating tens of thousands of unique miniature genetic Lego sets, each slightly different.
That's a good way to put it. And the experimental setup was, well, pretty ingenious. These thousands of synthetic constructs were first tagged with a unique genetic barcode.
Like a little label for each one.
Exactly. Then they were delivered all together en masse into mouse bone marrow progenitor cells. These are the cells that give rise to blood cells. And they used a special lentiviral system for this.
Ah, like a modified virus to shuttle the DNA in safely.
Yes, and one that's good at integrating the genetic material. Crucially, this system was designed very carefully to ensure single-copy integration.
Meaning each cell only got one of these synthetic enhancers, mostly.
That was the goal, yeah. It's critical for getting accurate measurements later. Then they used single-cell RNA sequencing, a really powerful technique.
To read the activity.
Right. To read each enhancer's activity by basically counting those unique barcodes across 8 different developmental stages of blood cells. So this allowed them to see precisely which synthetic enhancer was active in which cell type and how strongly. It gave them this incredibly detailed functional readout for every single one of their 64,400 custom sequences.
That is just a staggering amount of data. The scale is mind-boggling. Would have been impossible not too long ago.
Absolutely. Unimaginable just a decade ago.
Okay, so they have this mountain of data. How did they make sense of it all? How did they find the rules, the grammar?
This is where the computational analysis really shines. They needed a powerful tool. They used something called a Bayesian hierarchical model.
Okay, it sounds complex.
It is sophisticated, yeah. But think of it as a statistical tool that let them systematically untangle all the different factors. It literally dissected the contribution of everything. The order of the motifs, how far apart they were, even how certain pairs of motifs might cooperate or interfere.
So it could figure out which combinations worked best.
Exactly. And guess what they found? They found these relatively simple additive grammar blocks, combinations of motifs that, when put together, could predict over 70% of the enhancer's activity variance.
70%. That's huge. It suggests it's not just random chaos. There are actual rules.
Precisely. It's like finding the basic building blocks of a language, the Rosetta Stone for enhancer function in these cells. It shows the complexity is decipherable.
So they figured out the grammar. Did they then try to, write with it?
That's the crucial next step, the validation. And yes, they put it to the ultimate test. They trained a transformer neural network that's a type of AI really good at finding patterns in sequences, like language models.
Like the AI that powers chatbots and stuff.
Sort of, yeah. Similar underlying tech, but trained specifically on this vast data set of synthetic enhancers. And this network then successfully designed de novo enhancers.
Meaning completely new ones that weren't in the original library.
Exactly. Brand new sequences designed by the AI based on the rules it learned. And these AI-designed enhancers drove robust lineage-specific expression of GFP, that green fluorescent protein, a standard lab marker in primary human CD34E cells.
Human cells this time, not just mouse.
Yes, human hematopoietic stem cells, the ones that make all our blood. So this wasn't just understanding the rules, it was creating with purpose. Proving their understanding by building something new that worked just as predicted.
That is seriously cool. Compared to older studies looking at maybe a few hundred enhancers, this design, validate, iterate loop feels like a whole different ballgame.
Oh, completely. It elevates enhancer engineering to a new level, an AI-assisted, genuinely quantitative discipline.
It really feels like a game changer for how we approach gene regulation, moving from just observing to actually engineering.
It's a profound shift, yeah, especially given how tough this problem has been.
Now, you mentioned this involved different types of expertise. This kind of work doesn't happen in isolation, does it?
Definitely not. Science like this rarely happens in a vacuum. This work was anchored at the Broad Institute's Gene Regulation Observatory, sure, but it relied heavily on collaborations, deep ones. For instance, the Dana-Farber Cancer Institute provided those crucial primary human hematopoietic cell cultures. Those are notoriously tricky to work with.
Right, needs real specialized expertise.
Absolutely. And MIT's computer science and AI lab CSAIL, they helped optimize the transformer neural network architecture, made sure the AI could actually handle the sheer scale and complexity of the data.
And that data scale we talked about, 2 billion reads processed in 48 hours, that computational infrastructure must have been key.
Critical. It highlights the power of having these integrated computational resources, shared cloud pipelines. But it wasn't just the tech, They apparently had weekly data jams.
Data jams.
Yeah, bringing together the wet lab experimentalists, the ones doing the cell work, and the dry lab computational folks. It meant they could rapidly test hypotheses, keep everyone aligned. It sounds like a really dynamic environment.
It does. A fantastic ecosystem for discovery. Though I bet coordinating all that takes effort too.
Oh, I'm sure. Bridging those different scientific cultures always takes work, but the payoff, as you see here, can be immense. And it's worth mentioning specific people too, right? It wasn't just institutions.
Good point. Who are some key players?
Well, the paper highlights Priya Mehta as the lead computational biologist. She designed that complex motif grammar search space and actually wrote the Bayesian model code. Huge contribution. And on the experimental side, Diego Alvarez fine-tuned that lentiviral delivery system to minimize those positional effects we talked about.
Making sure the location didn't mess up the results.
Crucial. Super critical. To ensure activity differences were really due to the sequence itself. And the senior PI, Feng Zhang, apparently really pushed for that big scale up, from 6,000 to over 64,000 constructs, took a risk, betting on economies of scale, and his decision to open source the library afterwards, that's vital for the whole community to build on this.
Yeah, that's fantastic. Okay, so let's zoom out again. What does this all mean for the broader scientific landscape? What's the big technological significance?
Well, technologically, it gives us the largest experimentally validated enhancer dataset ever, basically, by far. And maybe even more crucially, it demonstrates that these transformer models, trained on synthetic data stuff we built, can actually outperform traditional methods that just correlate enhancers with genomic features.
So building from scratch can be more predictive than just observing nature.
In this context, yes, that's a really huge shift in approach. It suggests we can learn the rules better by actively designing and testing variations.
And what about the future of therapeutics? Where does this point?
I mean, this ability to really dial in cell specificity, to design enhancers that are active only where you want them, it moves the field significantly closer to designing truly custom regulatory circuits for gene therapy in vivo inside the body.
So going beyond just replacing a broken gene.
Exactly. It's about controlling gene expression levels and locations with exquisite precision. Now, of course, there are limitations we should acknowledge.
Right, always important.
Yeah. Much of the initial big screen was mouse-based, even though validation was in human cells. And those lentiviral integration sites, the chromatin context, can still have subtle effects. So translating these precise rules perfectly into human therapies inside the body, that'll need more work, further viral engineering, more context testing.
So it's a massive step, but not the final step.
Precisely. It's a breakthrough with clear next steps needed for clinical translation.
But even with those next steps, this study fundamentally shifts enhancer biology, doesn't it? From just describing what we see to being truly predictive.
Yeah.
And maybe even generative.
Absolutely. It lays down a foundational blueprint for what could become programmable gene switch therapeutics. Imagine, down the line, designing a genetic program to fight a disease with unparalleled accuracy, targeting only the bad cells, leaving the good ones totally alone.
It's an amazing vision. So you've heard how scientists are moving beyond just reading the book of life and actually learning how to write new chapters with incredible precision. This deep dive into designer enhancers really shows the power you get when you combine that scaled-up experimentation with cutting-edge AI.
Yeah, and this work really invites us to think, doesn't it? If we can truly program the very grammar of gene control, how might that fundamentally change our approach to things like chronic diseases, or maybe even regenerative medicine? What entirely new biological programs could we potentially write? And what ethical questions might pop up as we gain that kind of profound control over cell identity?
Definitely something profound to chew on, right? That's all the time we have for this deep dive. We hope you feel a little more well-informed and maybe a lot more curious about the future of genetic engineering. Until next time.